This video is for students enrolled in Bio 200 or Bio 201, Flow of Energy and Flow of Information labs at Pacific University. We're going to talk about how to convert a lab protocol to a flowchart. This is an important skill that you'll be asked to do many weeks during the semester. So we're going to start off looking at a generic protocol. This protocol is titled Pasteur's Classic Experiment on Spontaneous Generation. This is unrelated to anything we'll be doing in lab this semester. It's just an example of a protocol that we can convert to a flow chart. Protocols are a lot like recipes in cooking in that they'll tell you what ingredients you need to use and what order you need to combine them. What I want you to notice is that this protocol in particular, there are three separate parts. We're going to go from part 1 to part 2 to part 3, but they aren't necessarily linear. The first thing you're going to want to do when preparing a flowchart is to read through the protocol. I'll let you do this now. There is a document of this protocol posted to Moodle. The next thing we're going to do is illustrate it using visual images. So let's start off with part 1, preparation of a nutrient media for bacteria. Step 1 says, prepare a hay infusion by placing 5 grams of dry grass in a medium beaker. Okay, so we have to think about what are the most important parts of this step. So let's draw here a medium-sized beaker. Your drawings don't have to be fancy in any way. And what we're going to do with this beaker is prepare a hay infusion, so we're going to put 5 grams of dry grass. The important thing here is that you draw something that resembles grass. You put the amount, 5 grams, and you somehow show that this is going into the beaker. Step 2 is half fill the beaker with water. Leave room so the water will not boil over the top. So again, we're going to represent what's going into the beaker at this point. We're going to write water, or represent water in some way. And I'm going to show that we're going to fill the beaker half full. Notice that I'm using a bare minimum of writing here, only symbols or numbers representing what's happening, and instead communicating visually using arrows and pictures to show what we should be doing in this protocol. Step 3 is to place the beaker of hay and water on the hot plate. So we're going to be using a lot of things in lab that you may not know what they look like. And so if you know what a hot plate looks like, that's great. If you don't, you can literally just draw a box. A lot of lab equipment just looks like boxes. So I'm going to draw here, a hot plate on which to put our infusion. And you can label this if you want, hot plate. The next step, step 4, is to boil the hay and water until a dark tea is formed. Add water to the beaker if necessary. So this is something that you may want to represent by showing an arrow to represent time. So I'm going to draw an arrow here. And we're just going to say boil until dark. And then we can say add more water if necessary. Next, we'll move on to part 2. And what I want you to notice here is that, again, this is not a linear protocol. We haven't finished looking at what happens to our infusion. We're actually going to go on and prepare another part of this experiment. So here's part 2. I'm going to draw it in red now. So step 5 in this protocol is to sterilize 3 Erlenmeyer flasks with boiling water. So I'm going to draw here. And again, this isn't immediately following what we did in part 1, so I'm going to draw it down here. So this is what an Erlenmeyer flask looks like. We're going to use 3 of them, and we are going to sterilize in boiling water. Step 6 is to sterilize a solid rubber stopper and a 1-hole stopper by wiping with alcohol. So here's our solid stopper. Here is our stopper that has a hole in the middle. And again, we are going to sterilize these by wiping with alcohol. Step 7 is to cut a 16 centimeter piece of glass tubing with a file. The important thing here is that we're going to have a piece of glass tubing here that we know it's important that it be 16 centimeters long. Step 8 is then to bend the glass tubing into an S shape using a Bunsen burner. And again, we can cut out a little bit of this, but retain the important information, showing that we're going to bend using a Bunsen burner. Into an S shape. So here's our bent 16 centimeter glass tubing. The last step in part 2, step 9, is to insert the bent tubing into the 1-hole stopper. Be sure to lubricate the tube before attempting to insert it. So again, we're going to take this bent tube, we are going to insert it into that 1-hole stopper. So here's our tube, here's our stopper. And if you want to, you can write lubricate before inserting. And that's it for part 2. What you now see is preparing other materials that we're going to combine in part 3. Finally, we come to part 3, assembling the experiment. Because this is a separate part, I'm going to draw it in blue. So our next step, step 10, actually goes back to where we left off in part 1. Step 10 reads, after the infusion has been prepared and the glassware sterilized, remove the hay from the infusion with tongs and discard it in the trash can. Take care not to contaminate your sterilized equipment while assembling the apparatus. If the materials have been exposed to the air for more than 30 seconds or have been in contact with any surface, they must be re-sterilized. So that's quite a mouthful, but the real core important part of this step is that now we're going to remove the hay from the infusion with tongs, discard it in the trash can. So we're actually coming back up to what was happening at the end of part 1. So we have our hay infusion. It's been boiled to a dark tea color. And what's going to happen next is we're going to use tongs to remove the hay. So I'm going to show the hay coming out of our infusion, removed to the trash can. The second part of step 10 talks about re-sterilizing equipment. If you're not careful, you can either choose to leave this out, or if you want, come back over here and make a note that you're going to need to re-sterilize if necessary. All right. Step 11 is filter equal amounts of the infusion into each of the 2 flasks using a funnel and filter paper. Sterilize the funnel with steam. Sterilize the filter paper with UV light. So here's a little bit more of preparation that doesn't necessarily linearly follow from what we just did. So I'm going to put these sterilize the funnel, sterilize the filter paper over here with our other sterilization. So we're going to have a funnel here that we're going to need to sterilize with steam. And we're going to need our filter paper to be sterilized with UV light. Now we can go back to the first part of step 11, filter equal amounts of the infusion into each of the 2 flasks using a funnel and filter paper. So this is sort of like making coffee. You're going to have a filter or funnel here with your filter paper inside, and we're going to stick it into one of our sterilized Erlenmeyer flasks. And it says we're going to filter equal amounts into each of the 2 flasks. And so I'm actually going to draw out these 2 flasks here, as well as that funnel and the filter paper. And it's the infusion that we're going to be adding. So I'm going to draw an arrow here from that infusion showing it going into here. If you want to write a note to yourself, you can write equal amounts here and show that going into each of these Erlenmeyer flasks. Step 12 is to seal 1 sterile flask with a sterile solid rubber stopper and insert the sterile 1-hole stopper with bent glass tubing assembly into the second sterile flask. So essentially we're going to cap off both of these. I'm going to draw an arrow showing what's happening here. So again, we have a stopper, our solid stopper going into 1 flask here. And our S-shaped bent tubing stopper going into our other flask here. Step 13 is to place an equal amount of hay infusion into the sterile open control flask. Do not put any stoppers in the control flask. It must remain open to the air. Remember, we prepared 3 Erlenmeyer flasks, so here is going to be our 3rd one. We're going to draw it back up here because what we're doing is, again, adding now an equal amount into this flask here. And we're not going to put a stopper on it at all because it's going to remain open to the air. The final step, step 14, is examine the containers every day for 5 days. Look for evidence of life, such as cloudiness or the appearance of mold in the containers. So because this applies to all 3 flasks, I'm going to make these arrows converge, and I'm going to write examine every day for 5 days. Look for life. And this is how you convert a lab protocol into a flowchart. Again, I want you to notice that the words here are minimal but very necessary. And and that it's really crucial to read over the protocol before you start drawing the flowchart so that you have a sense of how you're going to lay it out on the page. If you want to look up anything you're going to want to draw or represent visually, and really get an overall sense about what you're going to be doing in lab the following week.